So we're going to start on the urinary system. We're going to go kind of quick because this part at the beginning is pretty easy. Okay? So urinary system, its overall function as a total system is to remove nitrogenous waste from the blood, okay, and eliminate it from the body. So it filters the blood of any broken down metabolic waste products, okay? Metabolic waste is produced from all cells of the body. It gets to the blood, and then it goes to the kidney and is removed, okay? It's eliminated in the form of urine, okay? And so the kidney does the work of filtering and removing the waste. The ureters, the urinary bladder, transport that waste, store it, respectively, and then remove it through the urethra, okay? Here is your urinary system. 2 kidneys, 2 ureters, bladder, and urethra. Okay? We're going to go through a little bit more of the anatomy of the kidney. All right, the kidney is bean-shaped. It has a hilum, little indentation on the medial side that allows for blood vessels to enter and exit, and also the ureter to exit. Okay? On top of the kidney, so the little top hat is the adrenal gland. Adrenal glands produce a number of hormones, and they're in the kidney for a reason, because they also is where aldosterone is produced. Okay, you'll notice that off the descending aorta is the renal artery. That's the blood vessel that goes to the kidney, and the one that leaves is the renal vein and enters the inferior vena cava. The ureters exit on either side and then attached to the bladder. The bladder is in the abdominal pelvic cavity. It can distend, all right, so it has a unique epithelial covering on the inside that allows it to stretch but still maintain a boundary between the urine that's being stored and the underlying cells. That is a transitional epithelium that stretches with that. Okay, there's a couple muscles and other voluntary muscles and involuntary muscles, smooth muscles that are present within the urinary bladder. Little email message, don't worry about it. That when there is voiding of the bladder, meaning expulsion of urine through the urethra, right, those contract and force urine out through the urethra, and there's both relaxation of sphincter muscles that allow that to occur. Okay, the kidney is what we call retroperitoneal. It's behind the peritoneum. That's why you see it on that right side there. Peritoneum is present, and then the ureters actually go into on the inside of the peritoneum and then down through the bladder. Okay, this is what it looks like on the cadaver. Pull back, you can see you have your kidney, hilum, blood vessels enter and exit. That's a ureter. And it looks like the bladder's been distended, or it's relaxed, right? Meaning it's closed after dehydration. Questions on the overall structure of the system? Okay, from the back side, you actually can see that the right kidney sits a little bit lower than the left kidney. There is some protection that's involved for the kidneys themselves. They are stuck in they are encapsulated. They have a fibrous capsule on the outside, and then outside of that is a fat pad, which helps cushion and protect them as well. And then you can see they sit underneath the ribs, the 11th and 12th ribs on either side. Okay? The kidneys are the working, functioning organs of the urinary system. And they have more than just one function in terms of producing urine, right? So with the production of urine, as an aside to that, it also controls water balance. Okay, you know, when you drink a lot of water, what happens? You need to go to the bathroom more, right? You are well hydrated, you produce more urine. Okay, you are less hydrated, you produce less urine. So it regulates water balance. Really, though, it's regulating blood volume. Okay? Because you're going to consume water, and that's going to be absorbed into the blood, right, from the digestive system, and that's going to increase your blood volume. More blood volume leads to increase in blood pressure, okay, because cardiac output is equal to stroke volume times heart rate, right? Yes, you agree with that? So if I increase blood volume by consuming water, what happens to my stroke volume? John, it goes up. Stroke volume goes up, cardiac output goes up, and if cardiac output goes up, what happens to blood pressure? Blood pressure is equal to cardiac output times peripheral resistance, right? So if my cardiac output goes up, what happens to blood pressure? It increases. And if blood pressure increases, the kidneys work to maintain blood pressure as well, okay? So all of that is related. So now these two things regulate water levels, blood volume, also helps regulate blood pressure. Those two things are very closely related, okay? And we've already talked about number 4, kidneys regulate red blood cell production, okay, because they produce erythropoietin. When there's a diminished capacity for the blood to bind oxygen, that is a stimulus for the production and release of erythropoietin, which will stimulate the production of more red blood cells, okay? The kidneys also regulate acid-base balance, and so we'll talk about that as part of chapter 25, right? And they'll work to excrete hydrogen ions, bicarbonate ions, and ammonia ions, right? H+, HCO3 negative, and H4. And then elimination of biologically active molecules, right? Hormones, drugs, right? Metabolites. So, one of the worst things that you can do is for your kidneys is to overuse of over-the-counter drugs, such as Advil or ibuprofen. Okay, my sister-in-law, okay, in her infinite wisdom, went in with a kidney problem, went into the hospital with a kidney problem last October. Okay, because she has a very chronic, painful shoulder that she was managing with taking 12 to 15 Advil a day. And so after a few months of doing this, her kidneys were not feeling so hot. Okay? So think about, that's kind of like all of that stuff, all of that Advil needs to be processed by the liver, and those metabolites need to be eliminated by the body. Okay? And so she put way too much pressure, way too much stress on her kidneys, and they were returning the favor by shutting down on her. Okay? And other molecules are involved with that. Here's the structure of the kidney cut in half. Cortex and medulla, the fibrous capsule on the outside. The cortex is where most of the neurons are found. Nephrons are found. Okay? Most of the nephrons are found. You have renal pyramids. These are little triangle things here. Okay, these are renal pyramids. This is the junction between those, what we call the cortical medullary junction. The area between the pyramids is the column. Okay, and then you have this area in the middle. That's the renal pelvis. At the end of the pyramid is what we call a minor calyx. That is where most urine is produced in the cortex, and then it's collected down the pyramids and into the minor calyx. Then that minor calyx fuses into a bigger area called the major calyx, eventually into the larger area before it leaves the kidney and the ureter called the renal pelvis. Okay, so you have the renal sinus contains minor and major calyces in the renal pelvis, so all of this right here. So when your dissections, okay, of your kidney in lab, when you cut that in half, all of that yellow stuff was in that yellow latex was where urine would be produced, or where it would be collected. Okay, so that would be all of where that yellow material would be called the renal sinus. All right, you see the renal artery, renal vein, and then as blood comes in from the renal artery, it separates and it breaks down, sends out in numerous vessels, eventually getting to the nephron, which we'll talk about next. Any questions on kidney anatomy up to here or urinary system anatomy up to here? No? Good. So, blood supply. So the function of the kidney is to filter the blood, so we have to have a very robust blood supply in order to do that. Okay? So if you follow this around from clockwise from starting over here at renal artery, all right, and follow it around, we will get to the point where we actually follow blood through the whole kidney. So renal artery, and then it breaks into two segmental arteries. Interlobar, the kidney is divided into lobes. Okay, so interlobar, it goes between the lobes. All right, and then arcuate, you'll see the arcuate one kind of runs over the renal pyramid. Okay, arcuate artery to an interlobular. All right, interlobar and interlobular are two different things. Interlobular artery, and then gets to a point where we are into the arterial blood, and there are two distinct arterioles that are important. The afferent arteriole, okay, afferent means incoming, and it's incoming into this structure called the glomerulus right here. Okay? The glomerulus. The glomerulus is a ball, a knot. So you can see it right here. All right here. It's a knot of capillaries. These capillaries are fenestrated. What does fenestrated capillaries, what do they do? What does that mean? They're leaky. Okay, they have pores on them and they're leaky. So any pressure that goes, any blood pressure that goes in through there, it's kind of like taking a garden hose and punching holes into it and then turning the faucet on. What's going to happen? Right? Water's going to come, fluid is going to come out. And that's what happens here, okay? So afferent arteriole into the glomerulus, and the glomerulus is the area, the spot where urine production begins, okay? That is the area where urine production starts. Okay, well, we're talking about blood here, so we'll come back to the glomerulus in a second. All right, so that is a capillary network in the glomerulus, all right? And blood runs through actually two capillary networks as it goes through the kidney. Number one is the glomerulus. Number two is down here, the peritubular capillaries. Okay, so we'll come back to peritubular capillaries in a second. So we go afferent arteriole, glomerulus, and then blood leaves again through an efferent arteriole. Efferent means outgoing. Okay? Why is it important that they're arterioles? What do you know about the function of arterioles as opposed to, why isn't that a venule that's leaving there? Why is it an arteriole? What do arterioles do that venules and arteries don't? And that's going to be very important for the function of the nephron and the kidney. What do arterioles control? Blood pressure. And how do they control blood pressure? What's the big thing that they do, that arterioles do to control blood pressure? They vasoconstrict and vasodilate. Okay? So the fact that these two vessels, incoming and outgoing, they are arterioles, what by their function, by their structure, what can they do? Vasoconstriction, vasodilation. Okay? So if an afferent arteriole constricts, less blood gets into the glomerulus. If an efferent arteriole constricts, more blood stays in the glomerulus longer. All right? It is a way of actually controlling blood that goes in and out of this functional unit. Okay? And it's very important when we start talking about what's happening at the glomerulus. Okay? So blood exits from the glomerulus from the efferent arteriole and then enters a secondary capillary network called the peritubular capillaries. There's a bunch of capillaries that are wound around the functioning unit of the kidney, the nephron. Okay? And there's actually another one, vasa recta is an extension of that peritubular capillary network. Okay? So it's an extension, and that's part of the nephron loop. So really you have this peritubular capillary network, which is around the proximal two parts of the nephron, and the vasa recta, which is around another part of the nephron. And then we get back from there, blood then enters the venule side, the interlobar vein, arcuate vein, interlobular vein, arcuate vein, interlobar vein, renal vein out of the kidney. Okay, so blood in, the pathway out. They are basically the same on the way in and on the way out, right? So if you looked at interlobar, arcuate, interlobular, that's the same as interlobar, arcuate, right, on the back side. There's no segmental veins. There's only segmental arteries. Okay, so that's the only part that's different there. Questions on blood flow through the kidney. So let's talk about the structure of the nephron. Okay, so blood comes in through an afferent arteriole and then ends up in your, what's that going to be then? John?
Glomerulus.
The glomerulus. That's okay. And then it exits via the what? Efferent arteriole. Okay? So blood is going to come in under a certain pressure and come into this glomerulus that has openings. Okay? And then that is going to be the first step in urine production. Those openings, that's going to push the pressure, the blood pressure is going to go in and push fluid out of the capillaries. And then it's going to be caught by this part of the nephron. Okay? This first part of the nephron is what we call Bowman's capsule. Okay? Bowman's capsule. And we're going to talk about, you're going to see a couple of different things. You're going to talk about renal tubules or renal corpuscles. A renal corpuscle is the glomerulus and the capsule. Okay? That's a renal corpuscle. From there, the next part, so this is really the first step in urine production. Blood comes in and pushes, what's going to end up happening is fluid's going to be pushed out of the glomerulus, and it's going to be caught by the capsule. And it's going to be taken up by the capsule, and then it's going to enter the nephron. Right? So the capsule's part of the nephron, and then we get this windy part. First part, this is called the proximal convoluted tubule. And then it enters this big loop. That's called the loop of Henle, or the loop of nephron. Okay? And then the fluid goes into a distal part called a distal convoluted tubule before it enters, a collecting duct. That's the basic structure of the nephron. Okay, so the nephron whole structure goes from Bowman's capsule to the collecting duct. Okay, and you have proximal convoluted tubule, loop of Henle, the distal convoluted tubule. Okay, most nephrons are out here in the cortex. Okay, so here's your renal cortex, shows your renal pyramid. You can see the collecting duct and where urine is going to collect. Most nephrons are what we call cortical nephrons. Okay, there's two types of nephrons. Number one, cortical. Okay, this one right here is a cortical nephron. Most of the nephron is in the cortex. Part of it extends into the medulla. Okay, and that's about 85% of the nephrons. The second one, nephron number 2, is Juxta medullary. Juxta means next to the medulla. This one over here is a juxtamedullary nephron. The loop of the juxtamedullary nephron extends deeper into the medulla. Okay? It has a longer loop and extends down. And that's about 15% of the nephrons in the kidney. What did I not include in this structure of the nephron in terms of blood vessels? What am I missing? What are those blood vessels that go around called, Case? What type of capillaries? Peritubular capillaries, right? So I didn't put in the peritubular capillary network, which would look something like that. And then it would end up towards the renal vein. Okay? Here's structure of the nephron from your book. Same thing, it's stretched out. You can see the afferent arteriole. There's a couple different parts. You have the vascular capsular space between the glomerulus and the capsule itself. All right, and then the proximal convoluted tubule and the loop of Henle and the distal convoluted tubule. Now this is also showing that there are a difference in the epithelium of the tubes that are surrounding the lumen. Okay, so a little bit thicker, thinner down here, and a little bit thick again. Okay, that's important because it allows for regulation of movement of solutes and water. All right, so the thicker the tubes, it's going to regulate what goes in and out easier. Right over here as well, the thin segment is going to allow different things to occur in and out. Okay? Questions on this structure? No? Awesome. Okay, here's another picture of the nephron, cortical nephron versus juxtamedullary. All right, you can actually see here part of, I want to mention, if you notice, if you were to follow this all the way around through this proximal convoluted tubule and the distal convoluted tubule, notice that part of this distal convoluted tubule runs back around and near the glomerulus. Okay? Part of that runs back, and that's important because it's an important structure that we're going to talk about. We're going to talk about in a minute. Okay? And that's what's called the juxtaglomerular apparatus. Okay? So it's a point of contact between the afferent arteriole and the distal convoluted tubule. So where it comes back before it enters a collecting duct, right here, okay, it goes in close proximity to the afferent arteriole and the efferent arteriole. And within that, there's a group of cells called the macula densa. They are modified epithelial cells, and they work to detect changes in sodium and chlorine ions within the fluid here. If this is too much salt, what's going to happen here? What's going to happen there? Okay, so they're important in recognizing this because then they're going to alter these. They're going to talk with these cells, okay, and help regulate the filtration that's occurring. Okay, so those macula densa cells they will signal granular cells within this apparatus to release renin, right? And we had Renin-Angiotensin-Aldosterone. What does that do? Remember we talked about that in regulating blood volume? The renin-angiotensin system, okay, will increase, right, will help regulate blood volume. We'll go over that again anyway. Right, so a couple different cells of that, the macula densa, the juxtaglomerular cells, that's over here, okay? So they're important. This is important in the regulation of arterial blood pressure. Because if you regulate blood pressure, you're also regulating blood volume, right? Those two things go hand in hand. Okay? So you can help regulate blood pressure by reducing blood volume, right? You agree with that statement? Yeah. Right? So if you reduce blood volume, you're going to end up eventually reducing stroke volume, which reduces cardiac output, which then will reduce blood pressure. Okay? It's kind of a stepwise progression. Those things are related. So this is important in the regulation of blood pressure. Okay? So questions on the structures that we've talked about of the nephron and the kidney. Okay? So what the nephron does, it produces urine. Okay? Starts as, well, really it's a collection of three processes that are involved with the production of urine. The first one is glomerular filtration. Okay? As blood comes into the glomerulus, it's under high pressure or higher pressure. Right? It has a higher pressure than normal capillaries. The glomerulus has more openings than normal capillaries, so more fluid gets pushed out than normal capillaries. Okay? More fluid out, and then it goes from there. Excuse. All right, so that filtration is the first step. All right, so that's what we call a filtrate. We'll talk about how that filtration occurs in a second. From there, that fluid that's collected in the capsule from the filtrate is then processed, moved through the nephron, and you have two other processes, tubular reabsorption and tubular secretion. Okay? So it happens within the tubes, proximal tubes, the loop of Henle, the proximal tube, and the distal tube. Okay? So originally, a lot of filtrate is produced, more than you end up getting rid of in terms of urine. Okay? But then reabsorption is we're going to take a lot of the stuff that we've put out past the filter back. Okay? So we're going to reabsorb stuff out of that filtrate. And then the last part is secretion. Sometimes that filter excludes things that need to get out of. So if there's anything excess or extra that we need to get rid of, we will secrete that by active transport, usually by active transport, into the tube or into the collecting duct, and so it can be expelled from the body. All right, so you have tubular reabsorption is out of the lumen, out from the tube back into the blood. Tubular secretion is from the blood into the tubes. Okay? So here's where those processes occur, basically. Right? Obviously, glomerular filtration occurs at the glomerulus. All right? So, and it is dependent upon the pressure of the blood flow coming into the capillaries. Okay? That's one way it's regulated. And then tubular reabsorption, tubular secretion have to do with the tubes of the nephron and the capillary network that surround the nephron, the peritubular capillaries. All right, so that's going to tubular reabsorption and tubular secretion, and then the vasa recta, also part of that peritubular capillary network, or an extension of it, I should say. Okay, with moving things in and out of the loop of Henle. Okay, so both tubular secretion and tubular reabsorption. The first step, filtration. Filtration is, by definition, is an exclusion based upon certain characteristics. In this case, the biggest, I guess, category or characteristic of exclusion is by size. Okay? It is by size. That means small things go through, big things do not. Okay? So the filter that we're talking about is actually made up of types of cells that surround the glomerulus called podocytes. Okay, so this is a podocyte, and it has these extensions that kind of is, the book describes it as an octopus-looking cell, right? So it has these legs that extend out, and from those legs are little small feet, pedicels as they're called. And those pedicels, they interweave with other cellular pedicels, kind of like your fingers like this, right? Interweave, and they form what's called filtration slits. Okay? And only materials that are small enough to pass through those slits, okay, are allowed out of the glomerulus, the glomerular capillaries. Okay? So it's based upon size. So the podocyte, in conjunction with the endothelium of the glomerulus, right, and the basement membrane of the capillary, make up what's called your filtration membrane. Okay? So this is what it looks like over here. Okay, so here on the left, you have the blood that's running through the glomerulus, and then you have your filtration membrane. So first part of that is the fenestrations, the openings. Okay, this is the capillary. The openings over here, the fenestrations within the within the glomerular capillary, allows stuff to go through. And then the next step, so you have, that's number one. Number two is the basement membrane, and number three are your filtration slits. Anything that doesn't fit through there or has a distinct charge doesn't fit through. Okay? Doesn't get pushed through. And you'll notice things on the left side don't get pushed through. They're too big. So you got large proteins, platelets, white blood cells, red blood cells. Those are too big to fit through the filtration slits. Okay? So what you end up with as blood goes through the glomerulus and all of this fluid is pushed through the filter, you end up with, on this side of the filter, something that resembles plasma. Okay? Plasma minus the proteins. Okay? Minus the proteins. Why not most of the proteins? Albumin, fibrinogen, all of those proteins that are present in plasma, they're too big. They're too big. Okay? You have very small amounts of proteins that get through there. Very, very small proteins can pass through. All right? So the filtrate we're talking about here, so is not urine yet. Okay? It's actually less concentrated than urine. All right, includes water, glucose, amino acids, ions, some urea, some hormones, vitamins, B and C, ketones, whatever can be found in the blood plasma without the proteins. Questions on the filtration membrane and what it does and how it works. This is, again, just by size. Size and to a much lesser degree charge. Okay, so some negative ions won't pass through. The filtration membrane is negatively charged. Okay? And this filtration membrane has been the inspiration for a lot of technical stuff that we use in biotech all the time. So when we make the fluid that we can grow cells in, we have to sterilize it. Okay? And so we run it through this filter, which is very small, so that bacteria can't fit through there. Right? So we've engineered this, and it's was inspired by this membrane that we have here. Okay? So as blood flows through these glomeruli, filtrate is formed. Roughly up to 180 liters of filtrate is produced per day. Okay? How much do you think you urinate in a day? Do you think you urinate 180 liters? Amanda says yes. Well, go get checked out. Okay? 180 liters, right? That's like, it's basically 4 liters to a gallon. Divide by 4, you're looking at, 40 gallons of urine per day. You're not an elephant. You're not an elephant. Okay? So, what's the point of this? What am I getting at? Most of this is reabsorbed. The filtrate goes through, but most of it, like almost greater than 90% of it gets reabsorbed. Okay? So, the filtration barrier excludes molecules based upon size and charge. And basically, you have 3 categories of molecules that stay in the blood at this point. Okay? Or you have 3 categories of molecules that are in the blood as it comes in. You got the molecules that are freely filtered, meaning that they pass through. That includes water, glucose, amino acids, ions, etc. Okay? You have things that are not filtered, they stay in the blood. White blood cells, red blood cells, platelets, large proteins, and then you get those things that are intermediate. Sometimes they get through, sometimes they don't. They're intermediate in size proteins. Okay? Normally, they do not get past. But in some cases, they do. Usually, it's indicative of some type of disease state. Okay? Whether it be too high blood pressure, whether it be attacking the cells that make up the podocytes that make up the filtration membrane or something, then you end up with protein in the urine, and that's indicative of some type of problem. Okay? So the filtrate resembles filtered plasma, right? And with certain solutes and minimal amounts of protein. Protein in the urine is called proteinuria, and it's indicative of, like I said, a problem with the kidneys or with the filter. So what drives this? What drives this? I've already said it a couple times. What drives this process of glomerular filtration? Pressure. Okay? Pressure. It's really involved with the pressure of the blood going into the glomerulus. Okay? And when we talked about capillaries, we talked about blood vessels, we talked about how much fluid gets out into the capillary, out of the capillary network. And there were 2 forces that were involved in that. The hydrostatic pressure, right? The pressure of blood flowing through the vessel, pushing fluid out on one end of the capillary, and on the other end was colloidal pressure pulling water back or fluid back in. Okay? Those 2 pressures are also present here. Okay? With an added pressure. So we have hydrostatic pressure is going to force, we said pressure out. Okay? Now, so HP out, or it's about 60 milligrams of mercury, right? So that's the measurement. That's hydrostatic pressure. That's the pressure of the blood flowing in. The fluid is going to push fluid out of the glomerulus. But at the same time, there's colloidal osmotic pressure that's pulling water back in as you're losing fluid. Some of it's being pulled back in because of osmotic regulation, osmosis on either side. Okay, and that's nothing new. That's what we talked about when we talked about capillaries, right? That's all those proteins now are stuck on one side, and then the osmosis doesn't like to be one way or the other. Higher amount of water on one side, and things get pushed back. So there's a slight amount of osmotic pressure back. Okay, and then finally, where is this going? Where is this fluid going? It's going out of the glomerulus into the capsule. Okay, thanks. Way to go, Dr. Vale-Cruz. You know everything. Thanks. Awesome. Okay, that goes into the capsule. Okay, But if there is fluid, too much fluid already in the capsule, that is now hydrostatic pressure that's pushing it back. Okay? So there's a little bit of hydrostatic pressure because blood's flowing through there, you're making filtrate all the time. Some of it is pushing back because this can't move too fast through the tube. Back towards the blood. Okay? So you're really balancing these two pressures. The pressure that's pushing fluid out of the vessel and the pressures that are pushing fluid back in. Okay? So the balance of that, so if you want to, you could tally it up. 60 minus the opposing pressures, 32 and 18, gives you your net filtration pressure. All right? In the glomerulus, it's about 10 milligrams of mercury. Okay? Bless you. So balance among these forces. Now I'm going to give you an example. Okay? And those of you who are in my lab, you've already heard this. Okay? My daughter was diagnosed with hydronephrosis in utero. Okay? Hydronephrosis means water in the kidneys. And it's a result of urinary reflux. When the bladder contracts, it pushes urine back up towards the kidney rather than its normal one-way exit through the urethra. Okay? And there's a whole different examples why that occurs. Okay? Or reason why that occurs. But when the urine goes back towards the ureter and back into the kidney, where does it go? It goes, and her urinary reflux was bad enough, it went all the way into the kidney. So her renal pelvis was much larger than it needed to be. What happens to the capsular hydrostatic pressure? Does it increase, decrease, or stay the same? What do you think? So here's her kidney, right? So here's her kidney and ureter. But now everything's getting forced back this way and back towards the collecting ducts. Okay? And so now if you look at the collecting duct, urine's getting pushed back this way. So urine's getting pushed back more than normal, against the normal flow, right? Now you have your distal convoluted tubule, loop of Henle, all right, and then the proximal convoluted tubule, eventually to the capsule. And urine is still getting pushed back against the normal flow. What happens to the pressure within the capsule? It goes up, right? So the capsular hydrostatic pressure is going to be higher. Okay? It's going to increase. Okay? Because of all of this is coming this way. Right? Everything's coming from the bladder, up the ureter, up the way it wasn't going originally, and that's going to increase hydrostatic pressure a lot. Okay? That caused damage to her kidney. Okay? It caused damage to the podocytes and the glomeruli that are in there. Okay? Now her kidney only works about 25 to 30%. Luckily, you have two, and you only need one. Okay? But that's an example of the hydrostatic pressure, right? So this is a pressure that's forcing stuff to be back to stay in the blood. Okay? Overall, what you end up with, the blood kind of goes through these glomeruli all the time, and you have a GFR, or glomerular filtration rate. Okay? This is a measurement of how much filtrate is produced. Roughly, it's 125 mils per minute in men, 115 mils per minute in females. If you calculate it out, you end up, and you can figure out that total plasma, all of your blood plasma, is filtered every 30 minutes. Do you go to so 125 mils per minute in 30 minutes, that would be what? 3 liters, right? Over 3 liters of filtrate that's produced. you'd have to be going to the bathroom every 30 minutes to get rid of that. Probably every 10 minutes to get rid of all of that. Okay? The bladder is moderately full at about 750 mils. Okay? With 3 liters of stuff in there, you might as well just catheterize yourself. Okay? So, gives you an idea about the reabsorption that occurs after this. Okay? And this GFR is one big way for us to control the production of urine. Okay? And that's influenced by the diameter of the afferent arteriole. All right? How much that will control how much blood comes in, comes out. We'll stop there. We'll pick up. We'll go over GFR and then tubular reabsorption and tubular secretion on Monday. And hopefully we will finish urinary by Wednesday. Look for those video lectures. All right, so you have them. Also,